So in this section, we're going to talk about how mutations happen at the molecule level and why that makes them become permanent, or how they become permanent, I should say, at the, in the DNA. So there are some mutations that are spontaneous. Those are the ones that we blame on DNA polymerase. So when we talk about spontaneous mutations, things that you could not prevent no matter how hard you tried, things that are naturally, things that happen in the cell, things that happen chemically that you cannot stop. And one of those things is called the tautomeric shift. If you've taken organic chemistry, you may remember tautomerization, especially in ring structures that have double bonds or double bonded oxygens, for example, in a ring structure, that you have these double bonds that can shift. And I'm not going to get into it. You may not have had organic chemistry, but the point is that when you have these ring structures and your nitrogenous bases do, that there are little shifts in the way the bonds exist, and it makes them kind of flicker between slightly different forms. Okay? And so you can have one form or you can have a slightly different form, and those are called tautomers. When a nitrogenous base does one of these little shifts, it can lead to what we call non-Watson and Crick base pairing. In other words, instead of T bonding to A, you have T bonding to G. All right? Or when you should have C bonding to G, but in this case you have C bonding to A. So it's the wrong pairing. And that's because really what's happening is right at the moment, you remember when DNA is replicated, the two strands come apart, and then DNA polymerase looks at the template strand and it brings in what it thinks is the complementary base. So if it sees an A, it should pull in a T. But in this case, when it sees it, if that base shifts right at that moment, then polymerase will misread which base that it actually is. And it will pull in the wrong base. And it won't even necessarily know that because the tautomer can form this weird base pairing with the wrong base. So in some ways it's not fair to blame the polymerase because it's just looking at what's there. And if it's a tautomer, the tautomer tricks the polymerase into putting the wrong base into the new strand. But we still blame the polymerase either way. So what happens is when you have these tautomeric shifts, and they're showing it right here, let's say you have a little tautomeric shift right there, the wrong base is added. So a T is paired with a G, which is wrong. And when that goes through another round of replication, they're showing those two strands being separated. Eventually, at least one of the chromatids ends up with a CG base pair at that position where the original was TA. I'm just showing you an example. And each of these is, this is one round of replication and this would be the next round. So it takes a couple cell divisions for it to become permanent. Now, by the way, when the mispair is inserted, there are repair systems that usually go in and correct that. But we're talking about a case where, the rare case where that mispairing does not get corrected. All right. Other replication errors are also considered spontaneous mutations. We talked about these repeated sections that can loop out. They're giving you a very simple one here, a repeated section of A's and T's can loop out. You've seen those 3 nucleotide repeat sections that can form the hairpins. So sometimes we call that polymerase slippage, although I don't know it's that the polymerase did anything wrong. It's really that the template strand folded back on itself. But again, we blame the polymerase. It's always the polymerase's fault, even though it sort of isn't. But the polymerase takes all the blame. So these kinds of insertions or deletions can come from during replication. Now here's one that we don't blame on polymerase. It's called unequal crossing over. It is spontaneous, meaning there's nothing you can do to prevent it. But you may remember in the bar I in the fruit flies, which was several chapters ago, and I put a little miniature here so you can remember this. If there are 2 regions that are very similar, during meiosis 1, in prophase 1, they're supposed to line up and cross over, and there's supposed to be an exchange of equal corresponding segments. But what if they don't line up quite right? What you can end up with is these duplications and deletions. So unequal crossing over results in insertions or deletions, and they can be any size. They can be quite large, like for the bar region it would be the whole gene, the whole section. It could be just a couple of base pairs. So that creates one of the homologs ends up with more, one of the homologs ends up with less. So it's a mistake during crossing over. So this is the one time we don't blame DNA polymerase. Now what about chemicals or other things that come into the body? Any chemical or radiation that comes into the body can cause mutations virtually in the same way. It's just that it's not a natural occurring chemical. It's a chemical coming from the outside in. When a mutation happens because of a chemical or because of radiation, we call those mutations induced. So spontaneous refers to mutations that happen due to natural events in the cell. Unequal crossing over is a mistake, but it's natural. Replication errors are natural. There's nothing that you could do to prevent them. Now, a chemical mutagen, in theory, you could avoid because these are coming in from the outside environment. So an example would be something called a base analog, or there's also nucleotide analogs. A base analog would just be something like the base portion, the nitrogenous base portion of a nucleotide, but it would be something that's a little bit chemically different than one of our standard nitrogenous bases, and it would misbehave and cause the mispairing during replication. So a base analog or a nucleotide analog. These are chemicals. So one of them is called 5-bromouracil. It's a uracil, but it has a bromine on it. So it's a kind of an altered base. And what it does is when it gets incorporated into DNA, in the next cycle, in the next cell cycle, it will cause a mispairing. So normally a uracil will base pair with, will act like a T, so it base pairs with an A in DNA. It'll act like a thymine. But then it might do a mispairing, and here's where the mispairing would be, because it should base pair with an A, but here it base paired with a G. And after a couple more rounds of replication, here they're showing, it takes a couple rounds of replication, but it can become permanent on some of the strands that now there's a GC base pair instead of what should be there, which is an AT. So you don't have to memorize. It's not anything specific. It's just, we're just saying that there are chemicals called base analogs which are incorporated into DNA, but then they misbehave when they themselves are looked at as part of the template. All right, here's some other chemicals that cause mispairing during replication. alkylating agents, also deamination, also hydroxylation. Don't memorize these. I like to talk about this one, though, because it's kind of interesting. So mustard gas, it's something that they used during World War I because it chemically burns your respiratory surfaces. But it turns out that it has sort of this other long-term effect. And what it does, it's an alkylating agent. So it would act a little bit like EMS, which is an alkylating agent. And what it does is it just adds these alkyl groups, all right? And it causes, whatever base that's added to, it causes it to mispair. So it's always about this mispairing, these chemicals that come in and mess up the nucleotides that are there, or they mimic the nucleotides, and then they misbehave right at that moment of replication so that polymerase puts the wrong base across from it on the new strand. So no need to memorize all these or like which one does what. But there are these chemicals that have these effects on DNA. And so in this case, it's a mutagen, a chemical that causes mutation, and it's because it causes, when it gets into your cells, it gets attached to, alkyl groups get attached to your nucleotides, and that makes them misbehave during replication. So people who got sprayed with mustard gas during World War I mean, a lot of people died in World War I, but the survivors, some of them developed cancer later, sort of that long-term effect of exposure to mustard gas. All right, another chemical, something called an acridine dye. An acridine dye has 3 ring structures fused together, which mimics, if you think about a base pair, a purine base has 2 ring structures and a pyrimidine base has 1 ring structure. So when you have a base pair, you have fundamentally 3 ring structures very close together. So these chemicals, these acridine dyes, mimic an entire base pair, and they insert into the double helix. So here's one, here's one, here's one, and here's one. So they're trying to show you a double helix, but when these acridine dyes insert into the double helix, and we call that intercalation. Intercalation means inserting into the double helix. It bends the double helix, and you can kind of see they're trying to show it's kind of bent. And anytime that the double helix is bent, the backbone is bent, that's going to make polymerase insert the wrong base. So again, polymerase is making a mistake because the backbone gets bent. It leads to mutation during replication. So now we're back to blaming polymerase. One acridine dye that we will talk about is ethidium bromide. It's a stain. It's a stain that is used on electrophoresis gel. So when you run DNA on an electrophoresis gel and you separate fragments according to size, you can stain it, you can soak it in a chemical that it's called ethidium bromide. But ethidium bromide is an intercalating agent, and then it fluoresces kind of an orange color. And then you can see the DNA bands, and it's because when it inserts into the double helix of the DNA that's on the gel, it glows. It glows under UV light. Sometimes in student labs we use a different stain, methylene blue, because ethidium bromide, not only does it intercalate into the DNA that's on your gel, if it gets onto your skin, it will intercalate into the DNA in your cells. And so it's a carcinogen. Anything that mutates DNA has a high chance of causing cancer. So when you handle ethidium bromide, you wear gloves. But usually in a student lab, we would use a different stain that's not a carcinogen, just for safety. But in any lab that I've worked in terms of research, we use the ethidium bromide. And now they have some that are not as toxic, they're not as carcinogenic as ethidium bromide, so they're starting to develop, or they have developed others. All right, what about UV? UV light comes from the sun. What it does is they're showing you just a little section of one strand of DNA. If you have two thymines next to each other, when the UV light hits, it creates covalent bonds directly between these rings of the thymine, the actual thymine base, which doesn't normally exist. There's not usually a bond directly between the pyrimidine rings. And so what that does is, look what it does to the backbone of the DNA. It bends it. It bends the backbone. And what did we say? When the DNA backbone is bent, when the molecule is bent, then during replication, polymerase will make mistakes. So polymerase is likely to make a mistake if there's a pyrimidine dimer. Now it can be, by the way, a pyrimidine dimer that refers to two pyrimidines next to each other. Usually it's two thymines. 95% of the time it's two thymines next to each other. So a lot of time we just say thymine dimers. But the other 5%, it can be two Cs, two cytosines next to each other, or a cytosine and a thymine next to each other. But usually it's two thymines. So I don't know why. I don't have an explanation for that, but they've done some research and that shows that. Okay, so it really doesn't matter how the mutation comes about. I mean, you can avoid UV light to some extent. You can avoid chemicals to a certain extent. But there are, like I said, about 2/3 of all mutations are spontaneous. So no matter how much you try to avoid these things, you're still not going to be 100% protected from mutations. Most mutations probably don't affect a large portion of your body, but the only ones that do would be the ones that start a cell to become cancerous. That's why we worry about that the most. So when we're looking at chemicals, though, that are going to be in food or in makeup or in other kinds of products, drugs, pharmaceutical drugs, if we were a company that was developing a product, we would want to know early on if that item was a mutagen. Because, I mean, to be practical, because of lawsuits and such. So a for-profit company wants to know if they spend a lot of money developing a product, they want to make sure it's not a mutagen. Because if it's something that's going to, potentially go into someone's body, if it causes mutation, then it's going to cause cancer, and there's going to be a lot of lawsuits. And from a healthcare perspective, it's, we don't want to hurt people. But from a business perspective, that's usually the aspect that they look at. But whatever the reason, you want to know how mutagenic any substance is. There's a test, a pretty well-known, kind of a standard test called the Ames test. And what it does is you can use it pretty easily. It's a screen. A screen is something that can be done cheap and easy and used on a large scale. And the basis of the Ames test is that you're going to start with bacteria that are auxotrophic, AUX, remember auxotrophs? And the ones they typically use are His-. That means bacteria that cannot make their own histidine. Histidine is an amino acid. And they take His- bacteria and they plate that on minimal media. And you'll remember that if a bacterium is auxotrophic, it cannot grow on minimal medium. So when you plate His- bacteria on minimal medium, nothing grows. Only His+ cells can grow on minimal medium. But what you do is you add the substance that you're testing. I'm calling it compound X. So it's whatever is being tested. You add that to the agar plate, which is how you plate out bacteria. The agar plate, the agar has the nutrients. You add compound X, whatever that thing is that you want to know about, into the agar plate, and then you spread the His- bacteria on top of that. Or sometimes compound X is put on a little piece of filter paper, like a little dot, almost like a hole puncher. Punch out a little piece of filter paper, soak it with your compound, and put that in the middle of where you put those bacteria. Now remember, if the bacteria remain His minus, then nothing will grow on the plate. So what you're looking for is there any growth on the plate? And those would represent reversions, or I guess they could represent suppression mutations that change the bacteria from having a His minus phenotype to a His plus. You're really only able to see phenotype. But if you see a lot of changes of the His minus cells to His plus, it means that there's a lot of mutation, or there's some level of mutation happening, and it's because of compound X. So this is the beginning of the Ames test. This is the methods. All right, you start with your His minus bacteria, and this is the control plate. This is just your His minus bacteria plated on minimal media, shown sort of as a pink color here. And then on this side, this is your His minus bacteria, and then some of your compound X has been added to the plate. Okay, so this is a control. Doesn't have compound X. And this one over here that I put an X on, it does have compound X. And both plates get some of that His minus bacteria. What will happen is, on the control plate, you may get a colony. They show one colony here, and you may get none. And that would represent just the spontaneous mutation rate that could happen in any cell. But if you have compound X, if this plate, when you grow it, you get 2, 3, 4, 5, 6, 7, 8, 9. You get 10 colonies. Then, and this one only had 1, then that you have to do some statistical tests. But if there's statistically significant number of colonies on the plate that contains compound X compared to the plate that did not contain compound X, that means that compound X is causing a significant amount of new mutations. And this picture here shows you the result when compound X is mutagenic. A lot of substances are not mutagenic, so a lot of times you have a control plate and you have your experimental plate, and they look pretty much the same. And if that happened, then your compound X is not a mutagen. One thing that happened when they first started doing this test is they would do the test, and they would have a compound that did not seem to be a mutagen, and then they allowed it to be put into products, and then later they realized that that was causing cancer. And so they finally figured out that some chemicals by themselves are not mutagens, but when they get into the body, the liver enzymes, the liver converts them into intermediate compounds, which are carcinogenic. And so now they add some liver enzymes also into the agar plates just to make sure. But in any case, they would be looking for, on the experimental plate where compound X is added, they would be looking for a rate of colonies, a rate of growth of the bacteria much higher than the plate that did not have the chemical. And so that would be an important thing to know before you develop a product out of this chemical, because it costs millions and millions of dollars to develop things.